Hi friends, today I wanted to discuss about the first year PU practical final examination, the pattern of it. As you know that you have 70 marks for your theory examination, of which mandatorily you have to score 21 marks to clear your examinations, and whatever marks you have in practicals, there is no pass or failure mark. Aggregate of theory and practicals you should be having 35, and with minimum score of 21 in the theory subject. So practicals is quite easy. Throughout the year you would have spent many weeks doing your practical work. First year PO practical is quite simple exercise. What you have to remember is in the practicals you have it for 30 marks, of which you have for practical 6 marks, and Viva Voce you have it for 4 marks. Throughout the year, whatever practical record you have completed and got it signed regularly, if it is duly submitted, you will be securing 6 marks. And 4 marks is for Viva Voce. You don't get an external examiner for the first PU examination. Most of the time, your subject teacher himself or herself would be your internal as well as external examiner. So they would be asking you 4 questions for the Viva Voce, and if you answer all the 4 questions properly, you get 4 marks. So very easily you can secure 10 marks out of 30 in practicals. The second part that you have to remember is conducting the practicals, for which you have 20 marks. The first question that you have there is micro preparations that you have to give of the given plant material A. Either you have to take a very thin section TS of monocot root or dicot root or monocot stem and dicot stem. Any one of this might be given. One trick for you to remember is if it is a stem, usually it would be greenish in nature, whereas the roots would be colorless, and that is one way that you can by physical observation shortlist whether it is stem or root material that you have got. Whatever small material you have been given, take thin sections of it, many thin sections of it, and using your brush transfer this thin section into the slide. That is one important aspect that you have to remember, so that the pith and other cellular details don't get crushed. So the thinnest one, which is almost transparent and is floating in the water, which almost looks like a very thin plastic material, like that sort of section, you have to take it up. See to it that they have uniformity in their section, because if they have one side of the section is thick and another side is thin, whenever you put the cover slip, there will be the formation of air gaps and you might be losing marks. You have to make a temporary slide, and for that you have to use, after staining, use the glycerin and make a temporary slide and focus it in the microscope. You have to call the examiner to check this slide. If they are quite happy with it, they will be giving you the full marks for the micro preparation of it. Then you have to, apart from that preparation and submission of the slide, you have it for 3 marks. Why you identified it to be such a section, for that you will be getting some marks. See, root, they would be having only 2 possibilities there, tetrarch or it might be polyarch material. So tetrarch to exarch in case of dicot root, and in case of monocot root it would be polyarch, that is many patches of xylem and phloem alternating with each other. Whereas in case of stem, what you can notice is if they have a regular arrangement of vascular bundles in a ring-like manner, and you can see a large pith, so I am very sure that that is going to be the dicot stem. But in case of monocot stem, the vascular bundles are scattered, that is attached to steel condition. And if you look into this vascular bundle which is scattered, they look like a joker face or a skeletal feature. That is very easy to identify it as monocot stem. So these are the possibilities. You have another choice. So usually they might be having a lottery method, wherein whatever chit you pick, accordingly you have to conduct the experiment. The second method is the demonstration of plasmolysis in the given material A, for which you have to show the plasmolysis, and you get 3 marks for the preparation and label diagram of the plasmolysed cell. You will be getting around 2 marks. usually they give you the Rio leaf, and you might be adding up the sucrose solution, so of 10 or 20%, and observe the plasmolysis. So plasmolysis, wherein the cytoplasm shrinks from the cell surface. And why we choose Rio leaf is they are naturally colored. So you will be taking the colored part of it, not the green epidermal peel, but the colored epidermal peel, so that it is very easy for you to identify. Once they have undergone plasmolysis, after plasmolysis, you can make a temporary slide and call the examiner to look into it. And for this, the label diagram, you get 2 marks. So that is very important. The third one is stomatal apparatus of the given material A. So generally they give the beetle leaf or Vinca rosea leaf or petunia leaf. So most of the dicot leaf, you should remember that the epidermal peel from should be from the lower epidermis, because if you remember the stomata in case of the dicot leaf or dorsiventral leaf is hypostomatic. So they will be found only in the lower epidermal peel. Once you have shown that and you have shown the staining and shown it to the examiners, you will be getting around 3 marks. And again, you have to draw the label diagram of the stomata, for which you are going to be given 2 marks. So that is how the first question carries 5 marks. You have 3 choices. By lottery method, you have to choose the question. The second question is given plant material B. You have to identify what is the plant material, the scientific name of the plant material. So see, you might be you have only 3 families: Fabaceae, Solanaceae, and Liliaceae. So Fabaceae, they might have given simple plant like Crotalaria gentiana or Pisum sativum. So then for the Solanaceae, Solanum tigrum. So simple material they would have given, or petunia. You have to identify to which family it belongs to. You have to write the scientific name. It is based on the Fabaceae, Solanaceae material that is available in that particular area that you get the plant materials. Whatever they have done in the practicals, the same plant material will be given even in your practicals from these 3 families. So Solanaceae and Liliaceae. So any of these 3 you might be given as the sample B. The scientific name of the material, it carries 1 mark. The name of the family, it would be 1 mark. And giving salient features of that family would fetch you 3 marks. So totally you have 5 marks. Or you can also, what you can do is, you can identify and classify the plant. So name of the family is 1 mark. Floral formula and floral diagram. Floral formula you get 1 mark. Floral diagram you get 2 marks. So scientific name, family name, 1 mark each. Floral formula 1 mark, and floral diagram you get 2 marks. This is the other choice that you have for sample B. So the third one is the C, where you have to do the biochemistry experiment. It can be glucose, sucrose, or starch or protein experiment. So the procedure, if you write it, you get 1 mark. The observation is 1 mark, and the result inference is 1 mark. An unknown sample is given. You have to do the experiment with the Benedict's reagent or biurate reagent or any of the reagent that have been used, and you write this experiment, procedure, observation, and inference or result. So each one of them fetches you 1 mark each, and if you are correct, you get 3 marks. Easily you can score it. So the identification of the experimental setup D. So there would be a physiological experiment. It can be a potato osmoscope or osmometer, transpirational pull, rate of anaerobic respiration. It can be paper chromatography, or differential rate of transpiration or imbibition. Any one of this experiment can be demonstrated. So you have to write the aim of the experiment for this experimental setup D, which you get 1 marks. The physiological principle concerned with that experiment, if you write, you get 2 marks. So totally you get 3 marks for this particular experimental setup D. Then you have identification and commenting of E. So identification, that is scientific name, you have to write. You will be getting 1 mark, and any 2 significant reasons for that identification, you will be getting 1 mark. So totally you have 2 marks. The slide or specimen, what they might give it for you is, it can be bacteria, oscillatorias, or spirogyra, or rhizopus or yeast, or it can be lichen, markensia, funereal Dryopteris, pinus, pea, maize, or modification of root or modification of stem, inflorescence or plant tissue. These materials you would have studied in your lab. Whatever material has been demonstrated and done to you, they will be giving the same material during the examination. So remember about this. So what you have to do is identification, that is scientific name of that given material E, and two reasons for the material to be identified, so and so, the significant reasons. So this is the third, identification and commenting of E, where you get 3 marks, 2 marks for it. Then identification and commenting of EF is there, wherein they might give, for identification it is 1 mark. Any two significant reasons you get 1 mark, that is half mark each for one reason. So I believe that when you are writing the reasons, write more than 2 or 3, so that even if you commit some mistakes in some of these significant reasons, you can score in the other points. So never be miserly in writing. You have a lot of time to do it. See, the F experiment, they might be giving it most of the zoological material, like it can be a slide or specimen like amoeba, or they might give you hydra, fasciola hepatica, ascaris, or ferretima, hiridinaria, that is leech, palimon, bombaxel, apis, that is the honeybee, Pilalasterius, Coleodon, labio, fishes, Ranalcalatus, Columbo, Orchidolagus, epithelial tissue, blood connective tissue, smear or smooth muscle fiber, striated muscle fiber, cardiac muscle fiber. Any one of these slides from the zoology part you might be getting, and you can easily score 2 marks provided you have done the correct identification. Second one is given valid reasons, 2 valid reasons. You can write more than 2 valid reasons, and that is how you can score marks. So you had for A 5 marks, B 5 marks, C was for 3 marks, so total it is 30. D was for 3 marks, so again it fetches you around 16 marks. Then E and F, 2 marks each, so totally you get 20 marks. The remaining you have to score through viva voce and practicals. So best of wishes for all the students who are taking up your practical examination. So be very neat and do the job neatly. You can score out of out marks in your practical examination. It is not going to be difficult at all. If you have benefited from this video, please like and subscribe for the channel, and also share it with your friends. Thank you all. Best wishes for your examination. Thank you.